Yeah, welcome back to my YouTube channel. Thank you for subscribing, thank you for liking and sharing our videos. Today we are going to look at red blood cell inclusion, the inclusion bodies. We are going to look at Holy-Jolly bodies, Haynes bodies, Pappenheimer bodies, basophilic stippling, Cabot rings, reticulofilamentous substances, hemoglobin H inclusions, C crystals, and malaria parasites. Let's first look at the Pappenheimer body. They are here. You can see there are these ones here. These ones here. This is a red blood cell, and for them they are remains of iron. So they are iron-containing bodies, usually found in at the peripheral. They always at the peripheral of the cell. You can see they always at the peripheral. They appear. You can get one or two in the cell, and they stain dense blue purple when you are using Wright's stain. And they are associated with splenectomy, hemolytic anemia, sideroblastic anemia, megaloblastic anemia, hemoglobinopathies. So these are the disease conditions. When you make a peripheral blood smear, you realize that they have the blood sample has the red blood cells have what we call Pappenheimer bodies. What we have to note here is that they are always located at the peripheral, and they are iron-containing bodies. These are the keywords we have to understand. Number two are the Holy-Jolly bodies. It is here. It is always a single. It appears as a single body, and they rarely appear two per cell. So you will always find one. They are small round compound of DNA. They are nuclear remnants. When these are the remains after the process of mitotic division. So they appear, they remain. And diseases associated include post-splenectomy, megaloblastic anemia, some hemolytic anemia, for example, sickle cell anemia, hereditary spherocytosis, erythrocytosis, stomatocytosis, thalassemia, also functional asplenia, and in severe anemia. You'll find that you'll get the Holy-Jolly bodies. What you need to remember is that they stain darker blue, darker purple with white stain. Another body is the Haynes bodies. The Haynes bodies here. This one is relatively larger. It is relatively larger than the Holy-Jolly bodies. So they are composed of denatured hemoglobins. So these are denatured hemoglobins. There are small round inclusions within the red blood cells, and they are caused by damage and denature to the hemoglobin. Components of red blood cells mostly by oxidative stress. Remember, there is what we call reactive. Okay. Now, also we have. Also, it can happen. They can appear due to genetic abnormalities in hemoglobin. These Haynes bodies are common in G6PD deficiency anemia, NADPH deficiency, also in chronic liver disease, and alpha thalassemia, also in hyposplenism and splenectomy. Another abnormality is the Cabot rings. The Cabot rings, as you can observe, it is 8-figured. It's like figure 8. It is a loop or figure 8 structure located within a red blood cell, and it is a microtubular remnant of mitotic tubules. during mitosis, those tubules, so these are remains. They are colored red purple, and the right is stain, and diseases associated with carbon rings include myelodysplastic syndrome, myeloblastic anemia, lead poisoning, heavy metal poisoning, liver disease. Another inclusion body is basophilic stippling. Basophilic stippling. You can see. They also called punctuate or dotted basophilia. They are round irregular shapes, granules of variable number. They actually always cover the whole cell. They are divided into two. They can be fine basophilic stippling in the polychromatophilia, and also we have the fine coarse ring, the coarse ring basophilia stippling, like in the lead poisoning and thalassemia. And they are stained by Reuters stain. Another inclusion body is the reticulofilamentous substance. They are artifactual aggregates of ribosomes. They are not visible on Reuters stain. So here a supravital stain must be used. They appear as deep blue reticulum network. They are these ones here. The arrow is in the normal reticulocyte. This is a normal reticulocyte. Lastly, we can also observe malarial parasites, and they are intracellular parasites in red blood cells. Malaria appears in different forms in the red blood cell. Thank you for watching.